Imagine you're looking at a war zone, and your primary defenders, the ones supposed to be protecting you, haven't just been defeated. They've been, like, completely captured and brainwashed.
Right.
And now they're actually building supply lines for the enemy.
Yeah, which is terrifying.
It is. And to make matters worse, their main weapons have been confiscated.
Yeah.
So in that scenario, if you're the general, you wouldn't want to just, drop a massive toxic bomb on the entire battlefield and hope you hit more enemies than allies.
No, you want to be strategic.
Exactly. You'd want to find a way to sneak those specific weapons right back into the hands of your brainwashed defenders. And that brings us to today's deep dive. We are unpacking some incredible new research today.
We really are. This study is called IRG1 Itaconate Rewires Macrophage and Lung Tumor Metabolism through G6PD inhibition.
Which is a mouthful, but our mission today is to look at exactly how lung cancer pulls off this insane cellular heist, and more importantly, how a single naturally occurring chemical in our bodies could be the key to dismantling the tumor's entire supply chain, literally from the inside out.
Yeah, it represents a really fundamental shift in strategy because, for a long time, the focus in oncology was purely on the rogue cancer cells themselves.
Yeah.
Just targeting the tumor.
Just kill the bad guys.
Right. But to truly grasp the mechanics of this heist, we have to look at the entire neighborhood, what we call the tumor microenvironment, or TME.
The TME, right.
Because a tumor isn't just an isolated ball of mutated cells. It is this highly complex, completely corrupt ecosystem. It recruits structural cells, it builds its own blood vessels, and critically, it dictates the behavior of the immune cells that get trapped inside it.
So let's focus on those immune cells, because they are the captured defenders in our war zone analogy. Specifically, I want to talk about macrophages because, I used to think of them as just the simple garbage disposals of the immune system, just Pac-Men gobbling up debris.
Which is a common way to think about them.
Yeah, but in this context, it's actually more accurate to view them as the tissue's local contractors, right? Normally, if there's a threat like, say, a condemned building or a rogue cancer cell, these contractors bring in the demolition crew.
Exactly.
But in the tumor microenvironment, they get bribed.
Bribed is honestly a great way to put it. Macrophages possess this incredible, but, very dangerous plasticity. It's a real yin and yang situation.
The yin and yang, yeah.
In a healthy scenario, they are aggressively pro-inflammatory. That's the anti-tumor state. They secrete toxic compounds to destroy threats and call in backup. But when they enter the tumor microenvironment, the cancer cells just bombard them with these chemical signals.
Like propaganda.
Basically, yeah.
Yeah.
And it coerces them into an anti-inflammatory state. They become what we call tumor-associated macrophages, or TAMs.
The TAMs.
Right. And instead of demolishing the cancer, these local contractors start secreting growth factors. They help the tumor build new blood vessels. They act as bouncers, suppressing other immune cells from entering the area.
So what actually controls that switch? Like what separates a demolition contractor from a corrupt builder working for the mob?
Well, the research points to a specific genetic switch, which is the IRG1 gene.
Okay.
And when this gene is activated, it produces a metabolite called itaconate.
Itaconate. Okay, so itaconate is the weapon we mentioned earlier.
Exactly. During a standard immune response, say your body detects, like a bacterial infection, macrophages rapidly ramp up their expression of the IRG1 gene. They produce just massive amounts of itaconate and use it to drive the severe inflammation needed to clear out the pathogens.
So pumping out itaconate is basically the biological hallmark of a macrophage that is ready for a fight.
Right, it's their sword.
Okay, so the researchers naturally wanted to know, well, what happens to this sword, this weapon, inside a lung cancer tumor? And to figure that out, they used something called spatial metabolomics.
Which is such a cool technique.
It's unbelievable. Specifically, they used a technique called MSI, and it's essentially a molecular satellite heat map.
That's a good analogy.
Because, normally you'd take a piece of tissue, grind it up, and look at the resulting chemical smoothie, right? But here, they shoot a laser at a microscopic slice of intact mouse lung tissue.
Right, maintaining the actual physical structure.
Exactly. It vaporizes the molecules pixel by pixel, allowing you to see exactly where specific chemicals are hiding within the architecture of the lung.
And the resulting map from that was just startling. In these mouse models, the healthy lung tissue surrounding the tumor was like glowing brightly with itaconate.
Tons of it.
Tons of it. But the exact moment the imaging crossed the border into the tumor itself, it became a dead zone.
Just a total blackout.
Complete blackout. The itaconate was mysteriously completely depleted inside the lung tumor.
Okay, so we're looking at this localized dead zone, but how is the tumor pulling that off? I mean, is the cancer actively destroying the itaconate once it's made? Or is it stopping the macrophages from producing it in the first place? Because those are two very different survival strategies.
They are, and the evidence points to suppression at the source. The tumor microenvironment is flooded with specific signaling molecules, cytokines, specifically one called TGF-beta, that essentially gag the macrophage.
So they can't speak up.
Right. These signals actively repress the IRG1 gene from turning on. So the tumor isn't just disarming the macrophages, it's physically preventing them from ever reaching into the armory to begin with.
Wow. And if you're listening to this, you're probably wondering just how crucial this blackout is to the cancer survival, right? Like, does it actually matter? And the researchers tested exactly that.
Right, using the knockout mice.
Yeah. They genetically engineered these mice ERG1 knockout mice to lack the IRG1 gene entirely. So these mice physically cannot produce itaconate anywhere in their bodies.
And the consequences of that were stark. When these knockout mice were subjected to lung cancer models, their tumors grew significantly larger and they multiplied much faster than in the wild-type regular mice.
Because there's no itaconate to stop them.
Exactly. And to prove that this was specifically a failure of the immune system and not just some quirk of the mouse's biology, the researchers performed a bone marrow transplant.
Oh, this part was fascinating.
It really is. They gave healthy mice the bone marrow, which are basically the immune cell factories, from the knockout mice that couldn't produce itaconate.
So the mouse has a perfectly normal body, normal lungs.
Right, but its immune cells specifically can't make this one metabolite. And even in that scenario, the lung tumors completely thrived.
Okay, so let's just do a quick 5-minute mini recap of where we are so far. Macrophages are the immune system's Pac-Men, but tumors brainwash them into becoming corrupt builders. Itaconate is the primary weapon they lose in this tumor environment. The cancer creates an itaconate-free dead zone, and without it, cancer just spreads wildly. So, here's a question for you listening to this. Think about that for a second. If the cancer's survival depends on creating this itaconate-free zone, what happens if we just force-feed itaconate back into the system?
Right. Let's just bypass the tumor suppression entirely.
Exactly. But there's a problem with that, right?
Yeah, there is. That is the logical intervention. But natural itaconate is a very polar molecule, meaning it really struggles to cross the fatty outer membranes of cells.
It bounces right off.
Pretty much. You can't just inject it and expect it to enter the cells where the actual fight is happening. So to get around this, the researchers tested a synthetic cell-permeable version of it called 4-octyl itaconate, or octyl ida.
Octyl ida, okay.
Yeah. By attaching a short carbon chain, an octyl group, the molecule becomes fat-soluble enough to just slip right through the cell membranes and get to work inside.
Okay, wait, I'm struggling with this part of the study a bit. You're saying this one modified molecule, this octyl ida, is somehow a silver bullet. Because the claim is that it starves the cancer cells while simultaneously waking the immune system back up. And, you know, biology is rarely that perfectly convenient. How is this single chemical distinguishing between a rogue, rapidly dividing cancer cell and a brainwashed macrophage?
That's the brilliant part. It isn't distinguishing between them at all.
Wait, really?
Really, it hits the exact same metabolic pathway in both types of cells.
Okay.
But because the cancer cell and the macrophage are relying on that pathway for completely different reasons, the resulting crisis affects them differently. And they proved this in vitro using human lung cancer cell lines like A549. At a dose of 250 micromolar, the octyl ida just completely stopped the cancer cells from proliferating.
And they tested it ex vivo too, right, on actual human tissue?
Yeah, on TDBCLS, which stands for tumor-derived precision-cut lung slices.
So it's living human tumor tissue, keeping the whole 3D architecture of cancer cells and healthy tissue intact.
Exactly. And it successfully reduced cancer cell division without killing the healthy surrounding cells.
Wow. Okay, so to understand how it does this without distinguishing the cells, we need to look at the cellular factory supply line, which is the pentose phosphate pathway, or the PPP.
Right, the PPP.
So normally a cell takes in sugar and burns it for energy, but a tumor is multiplying rapidly. It's trying to build physical mass, so it diverts that sugar into the PPP supply line.
It hijacks it.
Right. And this supply line gives the cancer factory two vital outputs. First, it produces mybose 5 phosphate. Those are the physical building blocks needed to construct new DNA and RNA.
Right, for cell division.
And second, it produces NADPH, and these act as antioxidant shields, because a cell multiplying that fast generates a massive amount of toxic cellular exhaust, and without those shields, the cancer cell will literally burn itself out from oxidative stress.
Exactly. It needs both the bricks and the shields to survive. But every cellular supply line has a bottleneck, right? An entrance that controls the flow. And for the PPP, that bottleneck is guarded by an enzyme called G6PD.
Okay, I want to introduce a little memory trigger here for the listener from our notes. Think of G6PD as the gatekeeper for 6-carbon proliferation defense, G6PD. This gatekeeper decides how much cellular sugar gets turned into DNA building blocks and antioxidant shields.
That's a great way to remember it.
Thanks. And the researchers discovered that Octyl IDA directly binds to this exact gatekeeper.
And they didn't just guess this. They proved it using a technique called 1-pentyl-13C2 glucose tracing.
Which is basically a radioactive sugar tracker.
Exactly. They could literally watch where the heavy sugar went inside the cell.
Yeah.
And when Octyl IDA was introduced, the sugar simply stopped entering the pentose phosphate pathway. The production of DNA blocks and antioxidant shields just plummeted.
Let's get into the mechanics of that block, because it's important to note it's a non-covalent bond. Right, it's not permanently destroying the enzyme, is it?
No, not at all. A covalent bond would be like pouring wet cement into the gatekeeper's lock. Permanent. Non-covalent is different. Think of it more like shoving a perfectly sized rubber wedge under a heavy door.
Okay.
It holds the door shut against the flow of sugar, but it's not a permanent chemical fusion. And we know this from the enzyme kinetics they ran. Octyl IDA dropped the Vmax, which is the maximum speed the enzyme can run, from 1.52 units down to 1.1.
So it significantly slowed the factory down.
Right. But it barely touched the CAM-M, the binding affinity, which went from 188 to 178 micromolar, meaning the enzyme isn't structurally ruined.
Oh, I see.
And through computer molecular docking models, they saw that Octyl IDA has a binding affinity of negative 5.9 kilocalories per mole for the active pocket of the G6PD enzyme.
It snaps right in.
It just snaps into place and perfectly jams the mechanism.
And for the cancer cell, the effect of that is catastrophic. Its supply chain is instantly severed, it has no building blocks to divide, and its shields drop, exposing it to lethal internal stress.
Which is why the healthy cells in that tissue slice survived, because healthy cells aren't desperately relying on a hyperactive PPP just to stay alive.
Right. Okay, let's do our second 5-minute mini recap. We introduced Octyl Itaconate, our fat-soluble weapon. It directly enters cells, binds to the G6PD gatekeeper, and shuts down the pentose phosphate pathway, essentially starving the tumor of DNA building blocks and antioxidant shields.
Exactly.
So question for you, the listener. If you are a cancer cell suddenly cut off from your antioxidant shields, how would you react?
Well, as we're about to see, the resulting stress changes absolutely everything. Which brings me to my earlier pushback. We have to look at what happens when that exact same rubber wedge is shoved into the G6PD gatekeeper of the brainwashed macrophages.
Right, the double agent effect.
Yeah, because the Octyl IDA is slipping into their membranes too. If their supply line gets shut down, why don't they just die alongside the cancer cells? It comes down to their motivation. Macrophages aren't aggressively trying to build new DNA to divide like cancer is. However, they are heavily relying on the other product of that PPP pathway, the NADPH, the antioxidant shields. The brainwashed pro-tumor macrophages absolutely need a constant supply of high G6PD activity to maintain their corrupt anti-inflammatory state. They need that pathway running hot to stay brainwashed.
Oh, wow. So when the wedge goes in and the gatekeeper is jammed, the macrophage's shields drop too.
Exactly.
What actually happens chemically to a macrophage when it suddenly loses its antioxidants?
It triggers a massive internal alarm bell, like a redox stress response. Without the shields, reactive oxygen species, that toxic cellular exhaust we mentioned, spike dangerously high inside the macrophage.
And that wakes them up.
It does. This intense stress forces the macrophage to upregulate a specific emergency response protein called HMOX1.
Ham oxygenase 1.
Right, HMOX1. And the sudden appearance of this protein acts as a hard reset for the cell's programming. The intense redox stress literally snaps the macrophage out of its anti-inflammatory trance.
Like waking up from a nightmare.
Exactly. It realizes something is very wrong, it drops its corrupt programming, and it violently shifts back into an aggressive, pro-inflammatory, anti-tumor fighter.
That is just a phenomenal biological pincer movement.
It's beautiful.
By jamming one specific gatekeeper, you cut off the DNA supply to the cancer cell, halting its growth, and at the exact same time, jamming that same gatekeeper in the brainwashed immune cell causes a stress spike that reboots its programming, forcing it to attack the starving cancer cell.
It's an incredibly elegant mechanism. But, the ultimate test of any mechanism is whether it translates to real human outcomes.
Right, the so what question. We've talked about knockout mice. We've talked about sliced human tissue in a lab.
Yeah.
Does this metabolic tug-of-war hold up when we look at breathing human patients?
It does. The clinical data strongly supports it. The researchers looked at the Cancer Genome Atlas, or TCGA,
Okay.
which holds just vast amounts of genetic and survival data for human cancer patients. And when they isolated human lung adenocarcinoma patients, a really clear pattern emerged regarding that G6PD gatekeeper.
What did they find?
Well, patients who naturally express unusually high levels of G6PD in their tumors face significantly worse overall survival rates. Their hazard ratio jumps up to 1.36.
Meaning a 36% higher risk of mortality.
Right, because their tumors have the factory supply line running at maximum capacity, constantly pumping out DNA blocks and shields. The cancer is just winning the metabolic arms race.
But there's a flip side to that clinical data, right? The researchers also looked at a protein called ABCG2. What exactly is that?
ABCG2 is a transporter. Think of it as a microscopic pump located on the surface of macrophages.
Okay.
Its job is to export the natural Itaconate that the macrophage produces out of the cell and into the surrounding tumor microenvironment.
So it's the delivery mechanism is how the macrophage actually gets the weapon onto the battlefield.
Exactly.
And what did the patient survival data show for this ABCG2 pump?
It showed the exact inverse of the G6PD data. Patients with high ADCG2 expression have improved outcomes. Their hazard ratio drops to 0.8.
Wow.
They survive longer, and this benefit is particularly pronounced in stage I, early-stage tumors.
Because their immune systems are naturally highly efficient at pumping itaconate straight into the battle, they are essentially doing naturally what the Octyl intervention does synthetically. They're flooding the zone and crushing the tumor's G6PD gatekeepers early on before the cancer can establish that dead zone.
Exactly. The clinical data perfectly mirrors the molecular mechanism.
Okay, time for our final mini recap before we wrap up. Shutting down the G6PD gatekeeper doesn't just starve tumors. It literally shocks pro-tumor macrophages back into being the good guys through redox stress. And real-world clinical data shows that patients whose bodies naturally pump out more itaconate via that ABCG2 transporter live significantly longer.
They do.
So here's a question for you listening. It really makes you wonder, right? How many other natural molecules is the tumor suppressing right now just to stay alive?
That's the big question. When you step back and synthesize all of this, IRG1 and itaconate represent a highly sophisticated dual-action metabolic switch. And it provides a blueprint for a completely new class of therapy.
Because for decades, the primary philosophy in oncology has basically been to invent highly toxic synthetic chemicals that blanket the entire body with destruction.
Right, hoping to kill the cancer slightly faster than you kill the patient.
It's the carpet bombing approach.
It is. But this research proves we can be strategic. By understanding and utilizing a molecule like itaconate, you can selectively cut off the cancer's metabolic supply lines while simultaneously waking up the immune system to finish the job. You're working in tandem with the body's innate biological programming.
It leaves you with a truly provocative thought to carry with you. We have spent decades and literally billions of dollars trying to invent the perfect alien chemical in a laboratory to cure cancer. But what if the ultimate weapon has been hidden inside our own cellular programming all along?
Yeah.
What if the future of oncology isn't about inventing new toxins, but simply mapping the exact natural metabolites that our immune system is being blocked from using and finding clever ways to deliver them right back to the front lines?
It's a completely different paradigm.
It really is. Think about that war zone we started with. We now know the confiscated weapon is itaconate. We know the enemy's weak point is the G6PD gatekeeper. And we're finally learning how to sneak the weapon back in. Thank you for joining us on this deep dive. Keep questioning the biology around you because the most powerful answers might just be hiding in plain sight at the microscopic level.